Hi, I'm Hannah, and genetic engineering sounds kind of scary, right? There's a lot of confusion about what it is. I mean, GMOs, designer babies, what's going on? Let's find out. Bacteria possess interchangeable genes on small circular pieces of DNA called plasmids. Bacteria exchange these genes in nature, when beneficial, often through a process called transformation. In the process of transformation, the bacteria inserts external DNA, most often in the form of a plasmid, allowing it to change its genotype and even phenotype. If there's a certain gene that we want to be expressed, we can cut it out and paste it into a specifically engineered plasmid. We can do this using restriction enzymes, which act as scissors and cut the DNA out. When two pieces of DNA are cut with the same exact restriction enzyme, they have complementary sticky ends and can be glued together. A promoter, a DNA sequence that initiates transcription and translation, is located upstream of the place where the gene has been placed. This DNA is called recombinant DNA. The plasmid is then inserted into the bacterium. Because bacteria reproduce asexually, their offspring are essentially clones, so if the parent contains the engineered plasmid with the special gene, their offspring will as well. And suddenly, there are millions of new bacteria with engineered DNA that have the ability to express the desired gene. Now, remember what genes do. Genes contain the instructions, in the form of DNA, that go on to produce proteins through the process of transcription and translation, which I have another video on if you want to learn more about those processes. Proteins are really important for regulating things in the cell and in your body in general. We can use this process to make mass amounts of proteins like insulin or human growth hormone. Before genetic engineering, we had to harvest proteins like insulin from pigs, which was far less efficient economically and in terms of effectiveness. The same method can be used to breed plant cells to make them immune to certain diseases, stay ripe longer, etc., allowing for an increase in crop yield. GMOs aren't fake, they're exactly what their name implies. They're just modified. A similar process can be used to treat genetic disorders in humans, except instead of a plasmid bacterial vector, a viral vector is often used, as viral DNA can integrate itself often permanently into human DNA. With further improvements in gene therapy, we have the potential to see a world without Parkinson's disease, immune deficiencies, and even cancer, to just name a few. Designer babies is the common way of referring to genetically engineering embryos to express certain traits. Ideally, this means replacing a harmful genetic mutation with a healthy alternative, not breeding a human super race. Although the technology exists today in the form of CRISPR and other methods, there are still a lot of risks associated with embryonic engineering. Thus, it's likely to be several decades before the first genetically engineered human is born. And that's genetic engineering in a nutshell. Thank you so much for watching.